Hello, this is Dr. Stephen Feldman, Professor of Dermatology at the Wake Forest Baptist Medical Center in Winston-Salem, North Carolina. I'll be discussing the publication, Acceptable Changes in Quality Attributes of Glycosylated Biopharmaceuticals by Martin Schiestl and colleagues. This study report was published in Nature Biotechnology. I selected this article because it provides more transparency anchoring the debate about acceptable changes in quality attributes. Data reveal physicochemical differences in each of the glycosylated recombinant therapeutic proteins in this study. This article highlights several different factors that may account for changes in quality attributes. Manufacturers are required to demonstrate the process change does not alter the clinical safety or efficacy of the biologic product based on principles of the comparability exercise regulated in guidelines. Well, let's look at the methods first. 3 major marketed glycosylated biopharmaceuticals were reviewed, analyzing the quality profiles of darbepoetin alpha, rituximab, and etanercept. The data revealed substantial alterations of the glycosylation profile for all tested products, most probably caused by changes in the manufacturing processes. The observed changes, however, in these studies predicted the reference medicine, and therefore its approved biosimilar do not alter the clinical profile and therefore are acceptable by the health authorities. Here are my thoughts and analysis of this study. This study provides a critical understanding of biologics, without which we cannot begin to understand biosimilars. Biologics are such complex glycoproteins that nobody can duplicate them. Many people hear that and think biosimilars cannot be trusted. But the principle that biologics are too complicated to duplicate means that even the innovator varies from batch to batch, as demonstrated in this study. Knowing we have accepted some degree of variation all along makes the idea of biosimilars a whole lot easier to digest. We will have far more data showing that the biosimilar is similar to the innovator product than we have had showing that different batches of the innovator product are similar. These findings make me realize that when I thought I was prescribing an innovator biologic that was a unique protein, I was actually prescribing a mixture of things that varied over time. That doesn't bother me or change my management in any way, because innovator biologics work and have worked great, consistently, wonderfully, despite whatever variation there's been. The results of this study make me very comfortable with the idea of using biosimilars, especially as those biosimilars will be tested to assure that they are similar and perform similarly to innovator biologics. This study is not informative, though, about all innovator biologics. Data on only 3 were provided. While this study is informative about the variation in the drugs, it is not informative about what clinical implications those variations have. Based on the clinical performance in my experience, I doubt the variation is meaningful, not clinically anyway. Moreover, the study does not tell us anything about the role of patient adherence and drug handling. I suspect the variation in how well patients care for and use the medication once they receive it dramatically swamps any differences in the batch-to-batch or innovator-to-biosimilar variation.